Hi everyone, welcome to another Plant Cell Technology stream. We are going to do a lot of work today. Let's take a look. Okay, so we're going to do a lot of work today with orchids. So I have a bucket full of orchids, and we are going to sterilize them on this live stream. We're going to sterilize them. We are going to place in biocouplers. So we're going to do that first, then we're going to pour some media, and then we are going to do some subculture while the plants is still sterilizing, and then at the end we're going to come back and do move the plants to the biocouplers. So I have my tools, my scissors here that I need to cool down because they've been in the sterilizer. So I just keep in the my shears in the sterilizer just because I want to make sure to keep it clean while I'm doing my cuts. It's very important because you don't want to transfer any diseases between the plants. So I just want to let them cool down for a second, and I want to start cutting plants. So I want to move the trash can here in front of me. Hello from Tennessee. Hello, hello. Yeah, so on this live stream we are going to do plants. Later, maybe in another live stream or in a video, we're going to do seeds. I do have some seeds for them, so for some orchids. So what I have done is I have 6 different types of orchids, and they all have a number. So I just label each of my jar with the number of the plant. So I know which one I need to take, so I don't get all mixed up because they are very important to keep the labels correct. So this is the first one. So I have my plant. Let me move my camera. Nice to catch live streams. Yes, of course. Do you review over my video suggestion? Can you please tell me or remind me what was the video suggestion? I want to get closer here. Let's see. We're looking at the trash right now. Looking at the trash because I want to cut the plants here. What was the video suggestion? Please let me know. Let's see. So here's the plant. So the first thing I want to do is any stick. There we go. Separate into like the two main plants. So here is one plant. So I want to cut the roots. I don't need the roots. So that is the plant. Now we have the second plant. There we go. So here's the stem. I honestly don't think I can get anything else from here. So I just want to discard this part. And this is the second plant. So I'm just going to cut. Oh, these shears need some sharpening. They're not great. Anyway, so I have cut the plant. Okay, so I have cut the plant. So the next thing I want to do is this plant, this is a vanda, this orchid. The hardest part to sterilize is because the way it grows, like the leaves, you need to make it to remove the leaves, because you need to have a good contact with the sterilizing solution. So if you leave if you don't remove the leaves, what happens is that the sterilizing solution will not go like let me try to point out. It will not go inside, like the in between the leaves. And what happens is that you will have issues with contamination because you may have issues with contamination because it was not the inside of this part wasn't clean. So what you need to do is remove the leaves, and the easiest way to do it, or the way I like to do it, is using my hands. So just peel them up with my fingers. I found that it's much easier to do it this way than using the scissors or the shears. So I just peeling all the leaves, and well, there we go. It just came out the whole thing. Let's see. I need the meristem, so I guess I can cut this part. You have to be very careful. So these plants also have nodes. So between each leaf there is like an internode. So you can. I think I still have the meristem here, but I also have another meristem here. So what I want to do is trim this other part. There we go. So I have my two parts of my plant. I'm going to place it in the jar. Okay, let's see. Can beneficial bacteria, mycorrhiza, fungi at the final stage when cutting in the jar will boost growth. So we're actually doing kind of experiments, something similar like that, but that's something that Shay is doing. I'm not very familiar with like mycorrhiza and fungi, so that's something that kind of little experiment that Shay is doing. So here's the second one. So I want to do the same. Just remove the leaves, do this carefully. There we go. Let me see if I can just cut this part like that. A little bit more. There we go. So here is my next plant. I don't need this whole thing, so I can just cut it there. And here is my other one, my other plant. I have been using 1 milligram per liter of BA and 0.1 milliliters per liter IBA, but I will get what I get is choose from the note and a lot of callus. With what plants are you working with? Because I know sometimes when you use a lot of BA, you get a lot of callus, so you may need to reduce the amount. All right, so I have the first one. So let's do the second set. So this is 2820. 2820. So I need to make sure to grab the correct jar. So I have 2820. And again with this one, so I will put my shears back in the sterilizer. And I'm going to grab the other set that I had, that is a nice cool, so I can cut using clean, sterile pair of shears. And this one is a little bit sharper, which is good. And the same thing, I just want to carefully remove the leaves of the plant. So this looks like very destructive because I mean it is. So from one plant, I'm basically taking one explant. Oop, I don't want to cut this too much because if I do it, I'm going to potentially damage the meristem. So I want to just cut, and this is going to be my explant. So there it goes. So that's plant number one. Let's do the second plant. So these are the same. So I have two plants of each. Let's put this out. Um, So it is very important when sterilizing the plants. Do not leave something like that. Like you need to remove everything. Make this as how they say like as aerodynamic as possible, where you don't have any like crevices. The sterilizing solution will not get into it. So you want to make it as smooth as possible. So there we go, and then remove the leaves. All right, let's see. So tomatoes, peppers, and some monstera. So maybe reduce the amount of BA. Then would be half a good start. Yeah, probably reduce the amount of BA. Usually, I don't have problems with callus with 1 milliliter. That's kind of odd. Oh, that was a really bad cut with these shears. Yeah, so usually 1 milliliter is, I don't really get issues with callus. You can also try something else like kinetic to see that works better for you. Let's see. All right, so I have my two explants for 2820. My shears in the sterilizer. Let me take this out. I need to let them cool down for a moment. I can also, to cool them down a little bit faster, I can spray them with a little bit of alcohol. That also want to help clean them up, but they are already sterile. Also thought about doing video community resources like local biohacker group and science spaces that can help beginners get into tissue culture and books online. So yeah, so we do a little bit of that. Maybe not like completely on YouTube, but we can do like shorts, like YouTube shorts or Instagram reels or like TikTok. 2819. Got 2819, jar 2819. Okay. People who are interested in plant tissue culture, yeah, of course. World community gardens help. Will community gardens help with a plant tissue culture? I don't know in like what context. Like, will community gardens will help with tissue culture? Hi everybody. All right, so new orchid. They're all bandas. I'm sorry. They're all new, all bandas, but they are like different variations. But I have a clean on my tools. And the first thing to do, get rid of all the roots. There we go. All right, so hormones for shoot induction, cytokinins. BA 0.5 to 10 milligrams. Wait, what? Okay. Okay, BA is 0.5 to 2 milligrams per liter. Oxins, NAA or IAA, and then 0.1 to 0.5. So let me tell you something. So for NAA, if you combine NAA and BAP, you have to be careful where you may have if you do high amounts for both, you may end up with a lot of callus growth, which, I mean, if that's what you're looking for, that's good. But if you are just looking for multiplication, you see, I just opened this part of the leaf and look all the dirt that came out of this plant. So that's why I try to remove as much. Look, there is a ton of dirt inside of there. So I try to remove as many leaves as I can, just so the sterilizing solution can penetrate that part of the plant. So yeah, and then IAA. So one of the things with IAA is great for rooting. The only thing is a natural cytokinin, which that means that it is not heat stable, and so well that means that if you're making media, you have to add the IAA after you have sterilized your media and the media has cooled down to like 55 degrees Celsius. So it is harder to use. So let's say for most beginners, I would recommend to use BA and IBA instead of using IAA, just because most beginners do not have a laminar flow hood, or maybe don't know how to filter sterilize hormones, or the plant growth regulator. So it's easier for them to add the everything when they're making the media. See, so this is the second orchid. Have I ever thought about tissue culture plant conservation? Yeah, I mean that's what we do. We do a lot of plant tissue culture and all plant conservation. Like we are not like a conservation lab. Here, Plant Cell is not a conservation lab. My role here at Plant Cell Technology is research and development of plant tissue culture products and new ways to get to grow plants in tissue culture. So I do a lot of that. So, but part of what we do, I mean, it applies to plant conservation. There we go. So that's the second plant. I have my two plants in the jar. Tools back in the sterilizer. Let's see, what is next is 2821. 2821. There we go. 2821. Here it is. Explore the role of tissue culture in preserving and engage plant species and restoring storing their habitat. Yeah, of course. I mean that's part of plant tissue culture that they use the conservation. Like I say, we are not like a conservation lab, but we do work with conservation labs as well. Not like part on part working with them, but if there is like any consultations that they may need help or stuff like that, we also have done that. Let's see, so This plant has a new shoot, so I would keep this shoot, the new shoot. I would try to keep it intact. Let's see. So let's remove the leaves. I'll be registering for your class. I'm a botanist in Iceland. Yeah, thank you. Yeah, definitely. We have the tissue culture master classes. Next one is not until July, so next month, pretty much a month from now, July 27 and 28. That's our next online tissue culture master class. So you can register now. So you can register at any moment. Let's see. So I'm trying to be extra careful with this plant because there is a shoot already on its way, and I'm trying to not damage it. Yeah, that's pretty cool. You build lamina flow hood. That's good. See, so another example, or what I was telling you, that what you want to remove the leaves is all this dirt that is just sitting in between the leaves. So let's cut this plant. Yeah, so the key for prepping next plant is to make them as smooth as possible. You don't want to have any crevices where dirt can stay still, where dirt may be sitting and the sterilizing solution can go in there. So just make it as smooth as possible. So that is the X plant. So grab the next plant. Have I ever tried tissue culture from medicinal plants? Yeah, I mean, we had tried a couple of medicinal plants. I had tried a lot of different plants. Like something that you guys don't see on like the background is I run a ton of experiments all the time, and with different plants. And hopefully, or something that we're thinking is like, well, every time where we do like tissue culture, we can probably go do it live. As it's like, I mean, why not? I mean, it's just content. It's like I had to use some tissue culture, and you guys kind of enjoy this. So if there is anything where like I can just show as much as I can. A lot of the times I'm doing like experiments where I don't know if the experiment will work. So I'm pretty open. So I always tell you like the media that I'm using and the sterilizing solutions and everything. But a lot of it, so, sometimes I'm working with experiments where I don't want to potentially. I have to be careful not to make them public. Not because I don't want to, it's just because they're experiments. So if they don't work, then you guys want to come back to me and say, hey, this didn't work. So I want to get blame on somebody's plans that die or something. I don't want that. So I just want to. So so I sometimes I will have to like just maybe don't mention like medias or sterilizing solution just because I'm running experiments. So I don't know if it's going to work or not. So all right. So this is my next plant. There we go. So that's the next band. Boom. All right, I have two more. I have 2701, 2701. 701. They sell pre-cut lumber kits online with everything you need for around $200. Wait, oh, hold on. I lost. What is the lumber kits for? I kind of lost track of that. Cryopreservation of plant germplasms explains the principle of cryopreservation and demonstrate techniques for preserving plant germplasm to go cryopreservation in tissue culture. So here's the thing. With cryopreservation, I don't think I can do that. And the reason is because I need liquid nitrogen. And that's kind of easy enough, can get liquid nitrogen. But we need liquid nitrogen tanks where they need to always, when we're talking about cryopreservation, it's cryopreservation. I mean, it's under like very, very low temperature. I mean, we need liquid nitrogen to keep it, we need to keep the plants constantly in liquid nitrogen. So it's not like you can keep the plants in the fridge. No, that's different. We're talking liquid nitrogen where you keep the plants for an indefinite amount of time. There are some resources where from University of Colorado, where they actually do have the videos on how to do the cryopreservation. I'll have to look them up. It's kind of hard to find them in YouTube. The videos are public on YouTube, but they are kind of hard to find. You have to really dig into them. I think it's easier to go directly into the University of Colorado website and find them from there. But they do have, I've been to their facility, to their preservation lab. And I mean, it's pretty incredible. They have massive liquid nitrogen storage tanks, but I mean, they had to run them all the time. And it is super, super expensive. So this is something that like only like preservation labs would do and not like commercial labs because it's just too expensive. But these are the guys that are preserving plants for forever, for thousands of years. So that's the cryopreservation. All right, I have my next plant. Explant is kind of long, so I think I'm just going to leave it there like that. So I read that you can use you can use 3M macropore tape. Yes, we do. We use that. That's to bend the containers. So yes, the 3M macropore will work definitely. But you have to be careful. I mean, when you pour in the media, depends. Like if you pour before sterilization, so you have your containers, and when you take it out of the pressure cooker, and then you have your containers, it doesn't matter if it's vented or non-vented, it's going to have a ton of condensation. You need to leave the container open in the hood to let it dry. But yeah, that's the option. Like whenever you take it out, just leave it open in the hood. If you don't have a hood, I mean, that makes it more difficult for you to control the condensation. But it definitely helps. Another way to reduce the I pull it up. Another way to reduce the condensation is to make sure to don't to reduce the light or keep it a little bit farther from the light. If you have the light is too strong, you're going to get a ton of condensation. Let's see. Hope I didn't damage this plant. I think the node's still here. The meristem is still here. But yeah, if you place the plants back in your like you see right here, I have my my racks, and then if for the plants that are like super close to the light, yes, you will see some condensation. It's actually not that bad. have a little, but yeah, you see the condensation. But with tissue culture, you're never going to have crystal clear containers. Unless you have vented containers and your light is about 18, 20 inches from the plants or the containers. All right, so last plant. Just to double check, is 2933. Yeah, 2933. Oh, for making the hood, yeah. For making the hood, a DIY hood. Yeah, you can definitely work on making your DIY hood. If you don't want to deal with like a kit and cutting and assemble it all yourself, we do have the the mini flow hood back in stock, so you can check it out. It's on the back on the website. The mini hood is back in stock, so we're ready to go. So that's another option too, where you can get starting tissue cutter with a mini hood. So let's clean the last plant. Does the condensation damage the plants? No, no, but depends on some of the plants. Like some plants are very susceptible to hyperhydration, where the plants will absorb too much water, and that extra condensation is definitely going to increase the chances of getting hyperhydration on some plants. So if you're working with a plant that is susceptible to hyperhydration, then you have to what I will start is to reduce the amount of light or move the plants farther from the light. Also check the temperature, that is not too hot. And what else? And use vented containers. So like you're using the micro portate, that helps. Now for the hole, to really help you, it has to be I mean, it doesn't have to be a huge hole, but you need to make it in a decent size where it's going to let some of the condensation out. Hello from North Carolina. Yes, that's awesome. North Carolina. I live in North Carolina for 6 months, like in the Wilmington area. Was nice. I love it. I enjoy it. Yeah, I'm doing tissue culture in some orchids. We have I have 3, 4 pots of seeds, green seeds, green pots. I'm sorry, that I got at the Orchid Festival in Florida. Somebody gave it to me, so it was pretty nice from her to give me those seeds. So that's something I want. I need to get that bit. I need to make that as a video where sterilizing those seeds and get those introduced in tissue culture. Because she gave those to me, and I promised her that I was going to make a video about it. I just haven't got to it yet. What species have you had the hardest time putting into tissue culture? I would say probably carnivorous plants. They are tough. They are difficult because they are very sensitive. Like some of the sundews, pinguiculas are very sensitive. Well, pinguiculas are easy, but some of the sundews are a bit more difficult to sterilize. Sarracenias are difficult to sterilize. Heliamphoras as well. All right, so I have my plants. So all of the bucket that I have full of plants is 90% now in these jars. It's on the jars right now. So I have my sterilizing solution, but first I actually want to clean my. This is non-sterile still, but I will clean my hands. Also, the laminar flow hood is off. Later on, I will turn it on. Well, actually, yeah, fairly soon I want to turn the laminar flow hood on because the rest of the things that we're going to be doing on the stream, they're going to be in sterile conditions. So I want to keep everything as clean as possible, and then I'll need the laminar flow hood to be on. So, sterilizing solution. And this is an experiment, so again, that's something that I'm just going to keep it to myself on the solution that I'm using. So let's see. I'll take a little bit more. There we go. So I have my sterilizing solution in. Very important, put the cap on and also shake the entire bottle, because if you leave it like this, I'm going to place this in the shaker, on the orbital shaker, but if you leave it like this and then the cap is still not in contact with the sterilizing solution, then you can potentially risk contamination. You need to sterilize the entire inside of the jar. So always flip the jar multiple times before placing it in the on the shaker. Do I remember good plant places in Wilmington, North Carolina? Well, yeah, of course. In Carolina Beach, there is basically sadly, one of the last habitats for Venus flytraps. So I remember that was a pretty cool experience. That's basically the plant that got me into tissue culture. That was the Venus flytrap. So to see it in nature, that was something pretty awesome. All right, so I want to move the plants to the shaker, and I'll be right back. All right, I'm back. Let's see. So those will go in the they will be sterilizing for an hour. So let me set a timer. Set a timer for 1 hour. So around 5:10, 5:15, we want to work with them. So I need to get this out of the way. So let me see. Just before I go to the next tab, I may answer a couple questions. How you thought about climate change? Well, I haven't really thought about doing about like climate change on tissue culture, how the tissue culture contribute to breeding climate resilient crops. Yeah, that's very important. With tissue culture, you can grow plants that are more resistant to certain, like not just the climate, but more disease resistant, more what else? Herbicide resistant. So more herbicide resistant, more insect resistant, more drought resistant. So that's something that is done in agriculture, so something that is great. So I know a lot of people are really anti like GMOs. Really, they always very anti GMO. I'm very neutral, so I try to consume organic as much as I can. But ever, I mean, that's my background, biotechnology. I have done like GMOs. I mean, I have done those myself. I can. That's what I studied. That's what I did, and I think they are important because in agriculture, I mean, we have a less farmland and less people who wants to farm. So farmers are getting very old. Young people, like let's say like me, don't want to be a farmer. So having crops that are more disease resistant, drought resistant, herbicide resistant are very important. So the kind of my opinion is like you are really into like organic. I mean, just consume organic products. That's perfectly fine. Or I mean, you can grow those yourself. That's probably the best case scenario. But when somebody is like really against and really protesting about it, about not doing anything, that's kind of where it kind of gets me a little bit. Where it's like, well, you are just kind of protesting, but you can just consume. I mean, just like I said, I'm very neutral, so I just kind of stay there. Yeah, exactly. All right, so I'm about to pour some media. So you give me a second. I'm going to change my camera. I want to move the camera to the other hood so you can see me pour media. Okay, so I'm changing camera. So there it goes. So the camera is now pointing at the other hood. I guess I won't need it. So the camera is pointing at the other hood. I'm about to pour some media over there. While I'm doing there, I won't be able to see the chat because the chat is on my screen right here, and I will be over there. So let me see. Let's see one more question. Yeah, pitcher plants, Venus flytrap, they're all protected, so that's good. But I mean, they still, sadly, they still um, a lot of poaching around. So biochar in your agar videos. Well, it's activated charcoal. What I've been using in some of the videos and some of the media lately. Yeah, so we have some media, some orchids. They do like some activated charcoal. So the media that I have right now, it doesn't have activated charcoal. So that's good. Sometimes working with activated charcoal can be kind of a bit of a pain because it is very dusty, and also cleaning your vessels, the all like vessels with all media that have charcoal is a lot harder to clean than use regular media. Regular media usually use the agar. You can use tap it on like the trash can and use usually everything comes out. For some reason, the media with charcoal just sticks to the glass or to the container a lot more, so it's a little bit harder to clean. But I mean that's I mean if the plants like activate charcoal, I mean that's what we need to use. All right, so I want to go ahead and um, start pouring some more media. So all right, so hopefully you can still hear me. You should be able to hear me, but let's see. I want to have my media. So my media just came out of the pressure cooker. All right, so here's my media. Here's my media. It just came out of the pressure cooker. Let me move this out of the way. So I spray this with alcohol. I'm going to put some new set of gloves. So this is carnivorous plant media. It has a gelen gum. Set this to the side. Spray my hands. So these are brand new containers, and something that I have learned is that when the containers are still sealed in the bags, they're still sterile. So I don't have to sterilize them. So I can just pour the media directly into the containers. I do have to be careful when I pour in the media, or when I take the containers out, not to get those contaminated. Also, I need to make sure there are no holes in the bags, because if there are holes, I mean, they potentially risk contamination. So I have put myself gloves, face mask. Again, I spray my hands one more time. Open the bag. So I'm doing 10 containers per liter. Alright, so the meat is pretty hot, so I use the glove to protect my hands. So I give it a good shake before I start pouring. One more spray with alcohol. And there it goes, about 100 milliliters into each cup. It has a magnetic stirrer on, so I have to be careful not to drop the magnetic stirrer on the container. There we go. I did pour too much on some where I don't have enough from the last one. Yep, I did 10. Just to make sure that I'm not doing 11 or 9. Alright, so there we go. So I'm in a laminar flow hood. If I want to avoid condensation, so that's something that we were just talking about just a moment ago, I will just leave the containers open there until they have cooled down, just for a moment, before I put the lids on. So that way I will avoid some of the condensation. I need to pour more media, so I want to go ahead and put the lids on. And I open it on the wrong way, which is unfortunately, I should have opened the other way around. Close the containers. Cover the lids. I will move the containers out of the way because I need to pour more media. I usually just leave it in here, but I need the space, so I just want to get it out of the way. All right, so now let me check there are more questions pretty quick. All right, let's see if there are any more questions. Let's see. Okay, no questions so far yet. All right, but yeah, if there are any more questions, you can just ask me. Like I said, right now I'm not going to be looking at the chat because I'm pouring the media over there. But I was just gonna keep going back and forth. So if there are any more questions, I can just come back and see if there is anything else. So let me switch camera again. There we go. All right, so this is the next liter of media. I know it does look overcooked. This media is a bit different, so that's why the color is different. Usually, if your media looks like that color is bad because the sugar has over caramelized, and that's not good. This media is different, so that's why the color looks like that. But if you are just making just regular MS media for houseplant, carnivorous plants, anything else, if the media looks like that, very orange, then that means that you overcook it. The sugar has caramelized, and that's not good. That's not good for the plant. So just make sure to, when you are doing, running the pressure cooker, make sure to always have a timer, so you know so you know when you need to turn the pressure cooker off. So spray my hands. So again, I'm going to do 10. Right, so I want to spray the bottle. Give it a good shake, and let's pour some media. This media, if you work on a research lab, looks like and smell like broth, like LB broth. So whoever works in a research lab kind of knows what I'm talking about. But that's very distinctive smell that this media has. It has like this color and that smell. There we go. Let's put the lids on. There we go. So have another liter of media. So again, I'm going to get them out of the hood so I can still pour the next media. But first, I want to take a little break to check the chat. Oh, let's see. All right, let's see what the chat says. What is the chat saying? Okay. I like your self-tilting biocoupler rack. It's a very neat. Do you think you can release a 3D print a file from the biocoupler jar connector? 3D file for the biocoupler jar connector. Oh, like the well, I'll grab the biocouplers later on. Let's see. What material you use? PLA plus? I don't. PLA plus. I will tell you. PLA. Oh, sorry. You're watching the hood. PLA plus will not survive autoclaving. I'll tell you. You have to use like polypropylene. ABS. I don't know about ABS. It will hold. I have not talked to him, to that YouTuber. Hydroponics and tissue culture, that's awesome. All right, let's pour the next little media. There we go. So we have another liter of media. Spray my hands. Okay. So you see, pouring media takes no time at all. It's pretty easy, pretty fast. Go. So after pouring the media, we're going to do some subculturing. So we're going to take some plants that are already growing in some biocouplers, and we're going to place into fresh media. We're doing that, and then after that, we move the orchids that are sterilizing right now on the orbital shaker to media. Let's see. Oh, by the way, actually, I need to turn on this laminar flow hood because we're going to do some subculturing. So it's going to get a little bit louder now. What can you do about browning in plant tissue culture? So for the browning in plant tissue culture is mostly due to phenols. So the plants start like leaking phenols, which if you don't know what phenols are, is kind of basically it's like blood on plants. That's kind of, I know it doesn't kind of the same thing, but it's kind of something that the plants release when they get like hurt or it's like, how to explain? It's like defense mechanism or something like that, where we get cut, we bleed blood. Plants bleed phenols when they get cut. So when you do tissue culture and then when you cut the plants, you are doing some stress to the plant. So it's like a stress release where how the phenols are released. Some plants will release more phenols than others. So there are some plants that it's very difficult to get rid of the phenols. And there are some other plants where you don't even notice them. But there are some plants that are very persistent. And I guess what we have found is ppm does help a lot with the phenols, with removing the phenols. But you have to change the media you have to change media. I wouldn't say constantly, but when you have the media, you make sure to add the ppm. So the phenols, you keep reducing the amount of phenols every time you are doing subculturing. For the most part, phenols are not like harmful, like super harmful, but they can stunt the growth of the plants if you have a excessive amount of phenols. If it's just a little bit, it's not really a big deal. But I know some plants you see that a lot. So you see that the plants they leak this, they look like bacteria, and then the plants they start turning like brown. Now another thing about browning in plants, it can be caused by ethylene gas. So that's it's called like sudden death on plants. When the plants they can be looking perfect, like healthy plants, and then very quickly, in like a week or 2 weeks, the plants die out of the sudden, and they turn brown, and also the media turns brown. And that's due to ethylene gas accumulation inside of the tissue culture container. So to get rid of that, you want to use vented containers, so to allow oxygen exchange. There we go. Alright, give me 1 second, guys. I just got a text. Okay. Alright. Let's see. So yeah, any more questions? Feel free to write it on the chat. I cannot see the chat at the moment, but I will check here in a second after I finish pouring this media. Oh, I'm so sorry. I left the face cam on instead of the other cam. All right. Well, you were looking the chair for for a few minutes. Uh, yeah, I just realized that. Yeah, would like to see some crops like wheat, corn, barley, triticale, oats. I have done actually a few of those, not on like camera, but I have done some of the tissue culture with some of those plants. Research from university, I work with wheat, switchgrass, rice, plants like that. All right. Have I ever tried tissue culturing aquatic plants? Yeah, I have done aquatic plant tissue culture. I have done aquatic plant tissue culture in the past. Um, it's something that I found like I haven't done in a long time, just because the plants that I work are like I'm working with whatever the demand is. So the demand right now is for like orchids and carnivorous plants and house plants and cannabis. So it's like I'm working with those plants right now and not a lot with aquatic because I know the demand's still there, but it's not like as it's not as high as some of these other plants. So, well, yeah, I definitely want to do some more aquatic plants. I know they are very hard to for the whenever you have them in culture, you get most of the time they will get contaminated. And I think I have an idea why or how to solve it. I just haven't got to it. What happened here? So they were supposed to be 50. Did I make a mistake? 1, 2, 3, 4, 5, 6, 7. No, it was 50. So I'm doing 10 containers per liter, but the sleeve that was supposed to be 50 per sleeve. 1, 2, 3, 4, 5, 6, 7. Have 47. So now I had to pour 47 on this other one. That's not like plant cell technology or anything. This is some other containers that I already have from back when I was living in Tennessee. So this is something that I bought from another company that I used to use these containers, and I still have some, so I'm using them. You can see they are not very consistent on the amount of containers that you get. So it's supposed to be 50, I got 47. And it happens more often than you think. A lot of times I get 49, 48. Rarely I get more than 50. So that's kind of the bad part. It's like if I don't get 50, then I will get like less, but never more. There we go. Hope you guys have enjoyed the empty chair cam for like 5 minutes or so. All right, so I'm done pouring this media. All right, so back here, I want to prep, get things ready. Want to do some soft culturing here. So have my tools. They're going on the sterilizer. There we go. Let me do a little bit of cleanup pretty quick. I'm also looking right now at the orchids. They actually look very good. I see that the sterilizing solution is doing its job. Um, it's doing its job. They look a little more white. So they were like darker green. They now look light green and they look more pale. That's for sure. So we have like, let's see, we have like 20, 30 minutes or so. Okay. So have my second camera as well. There we go, that looks good. All right, I'll be right back. I need to go grab the biocouplers and the media to move the plants too. And I will spray them, those biocouplers here in a moment. I know I need to spray everything, but I need to move the media first. All right, have some media. Let me position myself first. All right, so I will start by, let me move the biocouplers out of the hood first. I start with this an African violet in a biocoupler. This is 1 month of grow, just over a month of grow. The amount of grow that we have from how it started, I started with almost nothing to what it is right now is a lot. It's like very incredible results. But unfortunately, I need to use the biocouplers for some other experiments. So I need to take the plants out of the biocouplers and place in solid media. So clean some of the spray with alcohol, the containers. All right, let me check some questions before I get too far behind on the questions. So Can tissue culture be automated? I think we can automate some parts of tissue culture. I don't think it will ever be like fully automated, but we can make it really hands off. And something that the biocoupler does very well, this makes the process very easy. Where it's very easy to use the biocouplers and make it cuts a lot of labor. Because here you have to take from container to container and it takes some time. With this one it makes really easy, very easy. But fully automated, I know there are some companies that they have like this robot that has like the arm and then they just take the plants and place it into media. However, there's still a technician that does cut the plant and split them and put it on like a piece of paper, and then it goes into a conveyor and then the robot grabs the plant. So it's not like fully automated. It's still a lot of work. Still some work. The technician does most of the work, just splitting the plants, and to split the plants and remove every like dead leaves or everything. The robot cannot do that. Maybe sometime soon. Who knows? Do you prefer bleach or sodium dichloro for orchid plant sterilization? So actually, it's kind of funny because I'm using neither of those solutions right now to sterilize the orchids at the moment. I'm using something else. Again, it's an experiment, so whenever I have more data, more results, I make it public. But right now it's an experiment. I don't know if it's going to work, so I'm not going to tell you what I'm using. But for the most part, I think sodium dichloro, you have to leave it for like 24 hours and it gives you a broader window, sterilization window. With bleach, it can be difficult. You have very short sterilization window. If you already have a protocol that works for some orchids, for certain orchids, use it and it's fine. But for some of these plants, I mean, every single orchid can be different, like the size, the way they grow. So when that happens, I do prefer to use a longer, a milder solution, so like sodium dichloro for longer than using bleach for short amount of time. What is my favorite plant to work with in tissue culture? I think like something like Mexican pinguiculas, some of my favorite plants to work with. They just grow so well in tissue culture and they're fun. I love them, those plants. So there we go. So that's better view right there. So again, I want to clean the biocoupler, clean my hands. So I want to transfer some of these plants into media. So what I like to do is flip the plants so they are in the bottom jar with the media, and then open the jar. Open the container, and at this point, I can just take my forceps, which are sterile. They're actually still very hot, but I cool them down using the liquid from the biocoupler. And then I just place straight in the media. Straight in the media, right there. So I just want to apply some plants in there. And I just move this to the side. So second container. I know, right? That this alcohol container is, or the spray bottle is pretty cool. I lost this plant, unfortunately. It has this alcohol spray. It has a very fine mist, which I really like. Because we have another spray bottles that they use a lot. They spray a ton of alcohol, but this one kind of saves you a bunch of um, very fine mist, so you use less alcohol. It's more efficient, which I always like efficiency. So yeah, moving the plants out of the biocoupler because I need the biocoupler for other experiments. So like African violet is a plant that just grows really fast, and I already have enough of them, so I don't need I don't need the more African violets in the biocoupler. I will use it for some other plant that I need to propagate more of in a faster way. Just a reminder, if you want to learn more about plant tissue culture, please you subscribe to the channel. If you are not already subscribed, subscribe to the channel. We do like, I won't say every week, but we upload content on plant tissue culture very frequently. So we have a lot of good experiments coming up. So if you are not already subscribed, you subscribe to the channel. Also, use give a like to the stream, really helps a lot. And also any material, any tissue culture essentials, media, mini hood, plant growth regulators, gelling agents, can go to our plantcelltechnology.com, where you can get all your tissue culture essentials. I know we are out of MS Media at the moment. It's coming out soon. No, we're out of stock at the moment, but it's coming up, coming back soon. So African violet in new media. So I just want to move this out of the way. All right, next plant is philodendron. Oh, no, the QR code. Fluorescent dark form. I wonder how many people have tried to scan the QR code. All right, so again, same thing. I will any tips for African violet media? It's just houseplant media. The same media that I use for houseplant is the same media. It does great. So it is. Well, let me work on this plant first. Oh, there we go. See, that's also why I love the biocoupler. The plants, they split so easy. full MS media, um, 30 grams of sugar, 1 milliliter per liter of BAP, 0.1 milliliters per liter of IBA, or you can skip IBA all along. That's what I've been doing lately on my houseplant media. What else? 1 to 2 milliliters per liter of ppm, and that's it. Agar, 8 grams of agar or 3 grams of gellan gum, and pH is 5.8, and that's it. That's all the media. I wonder, like, and let me know in the chat, will anybody be interested on, let's say, my media recipe, or like the media that already has everything on it? So you basically just add water and pH, and that's it. I don't know if that's something that people will be interested or not. That's something we cannot be debating. Is like, should we just release like the media that has everything, plant regulators, everything already in there? So it will be like multiplication media. So it will be like my media, multiplication media. I don't know if that's something that people will be interested on it or not. Next is Alocasia black velvet variegated. Oh, this one is tight. There we go. There we go. Just keeping an eye on the timer for the orchids. It's getting close. They are almost done. So I think I just finished subculturing these plants and work on going back to and go back to the orchids. All right, so I need more media, so let me go grab more media. But let me spray this one first. So next is Sangonian milk confetti. Have you ever tried splicing two different plants together using a plant tissue culture? So I mean that's grafting. I mean at least that's what I'm thinking you are talking about, like grafting. You take two plants, different plants, where you take the it is done. This is done a lot with like trees, where you take a plant species that you know it has very good root system, and a plant, and the top of the plant that you know it has very good like fruits or like fruiting. So you combine the two, and then you get a better plant. So that's grafting. Um, I know I never had tried that on tissue culture. However, I do know back at the university we were trying to graft potatoes and soybeans. So we have like mixed results. We call them soy taters, soybeans and potato, soy taters. This plant does not look very happy. Okay. Last plant is Sangonium red spot tricolor. Oh, there we go. So this one pretty simple. Take my plant, which is great. Is I see a low lateral shoots. When I had this plant in gel media, they took forever to grow, and as soon as I place it in the biocoupler, it grew much better. However, unfortunately, I had to take it out of the biocoupler because I need them for other experiments. All right, so now this is the last one, which is another red spot leaf tricolor, so same plant. Oh, this one were two plants. Done. So this is done. All right, so I need to label these plants out, but I'll do that at the end, maybe on my own time. Let me get them out of the way. All right, and I'm going to take the orchids out of the shaker. Oh, they look great. Pretty happy how they look like. There we go. I hope they are not over sterilized. I think they're okay. I kick a biocoupler. Let's see. So I want to spray the plants, but look how great they look like. You see how white the plants look like? They look very clean. I really like how they look like. See again, looks very clean. So I use spraying all the jars with alcohol. So these are ready to go. Now I need to grab the biocouplers where I want to place the plants in. All right, so the first two biocouplers are here. Give it a good spray. All right, I also have my labels ready to go here. So as I work with the plant, I put my label. So make sure I don't mislabel everything, so I know everything is good. Can I explain hyperhydricity and phenos? So they are completely different things. Hydricity is when the plants absorb too much water. So I do have an example here. So before I get started, let me show you an example of hyperhydricity. Let me see. Where I saw one. Here, this one has a low of hyperhydration here. So this is hyperhydricity. Look how the plants look like. They look very pale. They look like very light green. Also, the leaves are super thin. They are super thin. The plants they have absorbed too much water, and these plants, although they are still growing, they're still alive, they will not survive outside of tissue culture. So these plants are done. So I won't be able to take any plants from here, unfortunately. But this is an experiment. So I run now you will say, why my plants look so bad? The reason why they look so bad is because I run a low experiment. So like I tell you, my job is to do research and development to try to find the best media to grow plants. So a lot of the times, like I go like different extremes because I need to test what works best. So this is very clear that this didn't work. So this media did not work for this plant, but I have some other media with different media composition that work great for this plant. But this is an example where it doesn't work. So it has a hyperhydration. That's a lot. This is also a non-vented container. So something that will help these plants to prevent this hyperhydration will be to have like a hole with a vent, something like vent like this, like the biocoupler. And phenos, I don't have a plant that has phenos at the moment. Um, Let me see. Maybe no, not really. No, I don't have a plant that has phenos at the moment. But you see, it's the browning of the media. The media start turning brown. It looks like bacteria. So the plants they leak kind of bacteria. So that's how they look. How it looks like. Are there any plant species you won't use in the biocoupler and find more success with solid media? I don't know about that yet. There are some plants where there are some plants that grow very fast. That the the biocoupler will benefit most the plants that are slow to grow than the plants that are fast to grow. But I have found that pretty much any plant that have put in the biocoupler does benefit from being in the biocoupler compared to solid media. But there are some plants that they just grow so fast that the difference is not as noticeable as, let's say, some plants that are really slow, where when I place this in the biocoupler, the results are just so much better compared to solid media. All right, I need to get started. I keep looking at the chat, but I also need to get started and move these plants because it has been an hour now. So let's get started with 2701. So I spray the biocoupler. I already have sprayed that. So open the biocoupler, the lid from this. Well, actually, before I forget, I almost forgot. I need to add. There is something else that I need to add to the biocoupler media. I need to do that in the sterile conditions. So I have my micropipette, sterile pipette tips. I need to add 2 milliliters into each. So make sure it stays clean. So I'm adding something that is not heat resistant. So that's why I'm adding this after the media has cooled down, or I'm doing it right now. But I need to do that to every single biocoupler. So look how easy is use the biocoupler. So I use one piece, second piece, and that's it. Close that up. Close this one. Sure they're tight but not over tight. Flip this one as well. Now we say it's 2701. So I look for a label 2701. There we go. And it has my QR code, everything, so that's my tracking system that I have. Perfect. So I move this out of the way. All right, so next to spray the biocouplers. Spray the jar. This is 2328. Open the biocouplers so I can add my other chemical that I need to add. So I grab a new pipette tip, a sterile pipette tip. And I add 2 milliliters to each jar. Next, then I just take the plants, see, I mean, this is so easy. This is very, very easy. Using the biocopper is very, very easy. Done. So we say it's 2328. Oh, 2328, that will be, let me take a look. I believe I have that under different name. Yeah, 2328 is this one. Double checking, make sure I don't mix the labeling. Make sure I have the correct labels. Perfect. Next to biocouplers. Are there any plant species? Oh yeah. I'm interested in learning about your labeling system. How are you? Well, I use an app. I'm still not kind of ready to tell you like what kind of app I'm using and everything. I'm still testing, but this is something that we needed, a system for us to keep track of all our plants and inventory. And this app makes things a lot easier for us to keep track, not only of the plants, but also experiments is great. So you just have the QR code. I use my phone or the iPad, take the scan it. It opens the app where I just add data to it. So all my data, every time I do something, like change media, add more plants, every month I take pictures as well. So I know like if I have a biocoupler that, or any plant, I used to track the growth of the plant. So every month I will take a picture and it shows the difference of when I started, and then after a month how they look like, and after 2 months, 3 months. I can do that very easy using the app. But that's something that is a pay app, where for us it's worth it, but for maybe for a person who is just getting into tuchoculture may not be necessary until you get like really big into, or like have a ton of different species. Well, it's very useful. I really like it, but again, it's something that we just still work in progress, getting used to it. But so far, it's very helpful. But yeah, it's always very important to keep good labels. So my labels have I'll show you my labels, but they have the full name of the plant, like they have the full name of the plant, the date, number, and I can have some more information, but also the app. I can make a much detailed label in the app, and I can just have the basics in my label. 2820. So there are only like a couple of details here in what you can read, so people can come over and they can see the name of the plant and when the date. But when you scan the code, and you can do, I mean, you can scan it, but it's not going to come anything because you don't have the app, you don't have my account. Will I have the media that I use, in every single date that I change media or I do anything to the plants or I just take pictures, it's in there. So every single detail. So that's this one. Twenty-eight, 21. Okay. So very simple. One plant into each jar. And although I cannot tell you like the sterilizing method that I'm using right now, and all the details and the media that I'm using right now, I'm actually very excited for this, because if this works, it's going to make tissue culture for many plants a lot easier. But like I said, right now I'm just testing. 28, 21. Well, I have done some preliminary tests using this method, and so far it looks pretty promising. So tissue culture is just getting easier for everybody, which is great. I know it used to be like a very difficult process that you will need like a really fancy lab or like a lot of like expensive lab equipment, you can do tissue culture from home now. And kind of part of my job is to finding ways to make tissue culture more accessible for everybody. So like the mini hood, we have the plant cell technology mini hood is back in stock, so anybody can do tissue culture from their own house now. 2819. All right, so I'm almost done here. I just got the last 2 yards. And also like the way the biocopper work, where it's just take the plant directly from the sterilizing solution and dump it directly in the biocopper. I mean, there is no easy way to do tissue culture than this. Like this is not bleach, I'm using something else. But, this one doesn't have a gasket. So it's makes tissue culture has been a lot easier because when using bleach, you have to sterilize, use the bleach to sterilize, and then you have to rinse it 3 times to remove all the bleach. Now you don't have to do that. Now you don't have to do that. So it makes a lot easier. Everything's a lot easier now. 19. Oh, this green holder thing. So this is for kitchen. It's a kitchen to keep your dripping tool or the dripping holder where you put your spoon or something. So it's a dripper thing. It's a silicone kitchen silicone utensil holder or something like that. That's how I call it. It is autoclave. I mean, it's silicone, so you can place this in the pressure cooker if you want to. It's pretty handy, it's pretty useful. Oh, I'm on my last set. All right, so 29.33. So you see, so there's I don't know, you've been watching the whole like stream, or this is going to be a video, so it's going to stay up on YouTube. But we have sterilized all the way from start to end orchids, and yeah, I mean they look pretty good. It was very easy. It was very easy, and they are going to be on the biocouplers now. So I'm going to be taking pictures of these plants pretty often on like a weekly basis, and hopefully we can get good results. Hopefully we can get a lot of plants because it's a very for a good project. So this is what we have. We have the plants. We have the plants now. So anything else? I think that's pretty much everything. So hope you have enjoyed the live stream. So I'm thinking maybe we're going to start doing some more frequent live streams now, starting soon. So hope you have enjoyed. I will see you in the next video. Let's see, any final things. Yep. So yeah, so expect some more orchid work pretty soon. Some more houseplants also coming up. Houseplant work coming up pretty soon. We have the tissue culture masterclass on July 27 and 28. So you are interested in learning more about plant tissue culture? Use you can get in the class. I forgot I completely lost my mind there. But yeah, you can get the class. I can see you the class. It's on Zoom. It's online, so you can attend the class from anywhere in the world. Anywhere in the world, we have over. I need to do the math, but I believe we have over 150 students, past students that have taken my class in the past. That's pretty amazing. A lot of great feedback from everybody. Yeah, I mean, I love doing these classes, so hopefully we'll start doing more. And we also have the webinars. So we do monthly webinars. So if there are any topics you guys may be interested on, you can leave those down in the comments, so we can potentially talk about some different plant tissue culture topics. So hope you have enjoyed it. I will see you